Hi guys, I just thought I'd quickly make that podcast I suggested in class today, just to reinforce this stuff. We need to come back to it. I won't take the full hour, I promise. I think 15 minutes will be more than enough. So last week we looked at that style lesson on DNA structure quite a bit. This is really just to reinforce that and hopefully to make connections to your projects. So here we go. The structure of DNA. There's that wonderful model using a modeling kit. So you've got nucleotides in model form in the middle and the phosphate and glucose groups around the outside making the backbone. So deoxyribonucleic acid, DNA, we find that in all living things. Now in eukaryotes, like our cells are, you find it within a nucleus, and so we have linear chromosomes, so it's all chopped up. But in bacteria, in prokaryotic cells, the DNA is free floating, so you tend to find it in one large loop within the cytosol. But the really important thing is that this code, this DNA, exists in all life forms. And so it occurred first. It's the egg, if you like, and the chicken came later. So without DNA, there's no life. We need to have this as a code for what happens inside a cell. As I suggested, in eukaryotes, we find all our chromosomes cut into little sections. So there's a karyotype of a human. There should be 46 chromosomes there if I could count them all. And each of those chromosomes has some genes on it, the little bit that codes for the production of a protein. And those proteins control what happens in your body all the time. When you're asleep, when you're awake, when you're doing things, when you're not doing things, constantly there's proteins being made, and you are taking information from your environment and deciding how to respond to it. That's your internal environment as well as the external environment. And we'll talk more about that when we look at nerves and hormones later this year. So there's our diagram of the backbone of DNA. So we've got that phosphate and sugar creating an external backbone, and then the base pairs adenine, thymine, guanine, cytosine bond together and cause those two strands of DNA to attach to each other, and that twists naturally into this shape called a double helix. There's a whole lot of detail there that you can look at, but I'll just pick out the eyes, the important bits. We also have a thing called RNA, and RNA is really important. So in things like our cells that are self-replicating, we can make copies of ourselves. You need DNA and RNA and the presence of a thing called a ribosome to be able to do that. So ribonucleic acid has more oxygen than deoxyribonucleic acid. There's only one oxygen different, basically. So again, they have a phosphate sugar background, although their sugar is not deoxygenated. We don't find thymine in RNA. You find a different base called uracil. So adenine and uracil bind together, and RNA exists as a single strand. And RNA is really very important. And a lot of people are now studying RNA more than DNA to see if they can do certain things to change diseases and control cancers and things. Now, the DNA in a human body codes for about 30,000 proteins. There are about 30,000 genes, each coding for a protein. Those proteins work together in large groups to create changes in your body. And of course, you're going through adolescence, and we talked about adolescence today in class and how many of these proteins, these hormones and other enzymes interact with each other on a regular basis and throw your body systems and rhythms out of whack quite a bit, including sleep patterns. So protein synthesis starts in the nucleus, and you have to unwind the DNA and just copy the section of DNA that codes for your protein. That gene section gets coded, and other parts aren't, so we delete those parts. And that is coded onto a thing called messenger RNA. It's able to leave the nucleus. The DNA can't. So the messenger RNA is just a messenger. It leaves the nucleus and comes out into the cytosol. In the cytosol, it goes to the ribosome. And so messenger RNA, which is a chemical, it attracts transfer RNAs, another chemical, and those transfer RNAs carry with them the amino acids, the building blocks of proteins. And at the ribosome, we end up with this polypeptide chain. So a protein is simply a polypeptide chain. That particular diagram shows a sulfur bond between the two cysteines. and there's cross-bonding shapes of protein. We'll talk about that in a little minute. Importantly, there's some stuff you need to know about genes and the way it's coded. So the information on a chromosome is coded for in triplets of nucleotides. So we talk about 3 nucleotides being read at a time, and that code doesn't overlap. So if you've read 3, you don't read them again. Next 3 is read. So the code's not overlapping. Code's universal. All living things have DNA, and we can take DNA from anywhere and put it anywhere. So we can move across species and between bacteria and all sorts of things. The codes tend to be redundant or degenerate because, as you saw the wheel in that class, the style class lesson last week, the same amino acid can be coded for with different triplets. The information tells you which amino acids to put together. The information's unambiguous, and you get a start and a stop instruction. And the start instruction, of course, comes with a methionine attached. When you've got the protein, when we've come out of the ribosome, we've got this polypeptide chain, and it's sort of the primary structure of a protein. So it's the first stage. Nothing's really happened yet. It's not useful in any way. It's going to be shaped up. So that moves into the endoplasmic reticulum, and in the endoplasmic reticulum, we start developing the secondary and tertiary structures. So it starts by creating these helixes, these alpha helixes, and we get these sheets bending up, and we get pleats, what we call beta pleated sheets, and that causes these random coils to appear as well, because all these amino acids have ability to attract each other, but also to repel each other. And so if they're attractive, they move together, and if they repel, they move apart. That starts to change the shape of this protein. And then you get a thing like this. This is a computer simulation, but you can see the alpha coils, the big flat bits are pleated sheets, and the whole thing's wrapped around itself. And within that, there's an active site, a place where the protein, in this case, the protein attaches to another chemical and creates a chemical reaction. So it's that binding site that's really important. Now, the tertiary structures also can develop within the endoplasmic reticulum, and then if it's going to leave the cell, or if it's going to be made into a larger protein, it moves into the Golgi apparatus, and we get this quaternary structure. So in the case of the one on the right, the hemoglobin, red blood cells carry hemoglobin for binding with oxygen and carbon dioxide. That's actually 2 chains, 2 proteins joined together, the alpha chain and the beta chain, and that creates a binding site for oxygen and carbon dioxide. The collagen's another one in your hair. It is a quaternary structure. It's a number of proteins joined together, and that happens in the Golgi apparatus. Proteins are critical for us. You know, we are essentially based on the operation of our proteins. So enzymes control our cellular processes. We use enzymes to digest our food. We use enzymes to control things in our metabolism. We have proteins for structural support. We have proteins that allow us to move. So our muscles have many proteins in them. That's one of the things that plants lack, is the number of proteins that we have. And so their ability to move is greatly restricted. We use proteins for transport things. So we have protein transport channels across membranes and things. We also use it for cell recognition. So our immune system recognizes the self-markers that a cell puts out, and they're proteins. And those proteins are coded for by your DNA. So your DNA marks your cells as yours, and your immune system therefore doesn't attack them. Cells also talk to each other in other ways. So this signal transduction is when cells signal between each other and coordinate their functions. Hormones are used to coordinate functions of whole organ systems. And of course, as adolescents, you're having a lovely time with hormones racing on your body, developing you into fully grown adults. And that's causing all sorts of problems. Potentially. Not everyone goes through it. Some people go through it calmly, some people go through it, have a really hard time, and we all do it differently, somewhere in between. We talked about defense markers. And that is our short little chapter on DNA structure. And it will be very important to you when you come to think about your topic and how your genetic dilemma is influenced by the manner in which the genes involved are in fact expressed. So I hope that helps. And tomorrow we'll have a lovely prac, and we'll spend some time with that.